Chapter 3: The venous drainage of the spinal cord and brain. Disclaimer: I do not own the rights to the images or information. This was purely made for the purpose of studying for boards. The venous sinuses of the neural axis of two components, namely the spinal system and the cranial system. The spinal system drains the spinal cord and the vertebra. Cranial system drains the brain and certain extracranial regions. The two components join to form one continuous system of venous sinuses, in which valves are poorly developed and scarce or even absent altogether. Transverse connections from the left to the right side of the system of sinuses are common. They include the midline cavernous sinus, the transverse sinus, and the anastomosis of the spinal system across the midline. All of the venous sinuses are outside the dura mater. In the head they are sandwiched between the dura mater and the periosteum. In the vertebral canal they are in the epidural space. Spinal system of the venous sinuses. The main component is a pair of longitudinal spinal sinuses running parallel with each other along the whole length of the vertebral canal. They lie on the ventral floor of the vertebral canal. The valves are absent or poorly developed. The transverse connection from one side to the other occur erratically at the junction between the vertebrae. The left and right longitudinal spinal sinuses are sometimes called the left and right vertebral sinuses. In the nomino-anatomic veterinaria they are named the internal vertebral plexus. Connections to the cranial system of sinuses. At the foramen magnum there is direct continuity end to end of the longitudinal spinal sinuses with a connecting system of cranial sinuses. Spinal sinuses drain the whole length of the spinal cord and the cauda equina. The veins of the spinal cord empty into the ventral spinal vein, which accompanies the ventral spinal artery along the ventral fissure of the spinal cord. Other veins drain from the spinal cord into the network of veins lying on the surface of the cord. Veins accompanying the roots of the spinal nerves drain the ventral vein of the venous network into the longitude of spinal sinuses. The spinal sinuses to drain the vertebral bodies including their hematopoietic tissue. Drainage of the spinal sinuses into systemic circulation. The longitudinal spinal sinuses drain by intervertebral veins. These are segmental and escape from the vertebral canal by passing through the intervertebral foramina alongside the spinal nerves. In the neck they empty into the vertebral veins. In the thorax that drain into the azygos veins. In the abdomen they discharge mainly into the caudal vena cava and also into the azygos veins. Valves direct the flow away from the spinal cord, but distension of these into vertebral vein and apparently render their valves incompetent. The cranial system of the venous sinuses. Mentioned on the next picture. Cranial system of venous sinuses can be subdivided into three of the sinuses. There's the dorsal system of sinuses, the ventral system of sinuses, and the connecting system of sinuses. The dorsal system of sinuses drains the dorsal areas of the forebrain by the dorsal cerebral veins. These areas include most of the cortex of the cerebral hemispheres. The dorsal system of sinuses also drains deep dorsal parts of the forebrain by the great cerebral vein. The eventual system of sinuses drains the eventual areas of the forebrain by the ventral cerebral veins in the face, nasal cavity, orbit, and upper teeth. The orbital drainage includes the angularis oculi vein, which drains the skin in the region of the upper eyelid and is used for cavernous sinus monography. This vein, which is boundless, empties at one end of the facial vein and at the other end into the dorsal external ophthalmic vein and thence into the cavernous sinus. The nasal drainage is important in protecting the brain against a rise in body temperature. The connecting system consists of a paired connecting sinus known as the sigmoid sinus. Deconnecting sinus is a focal point of the cranial venous sinuses. It receives the dorsal system via the transverse sinus and the ventral system via the control sinuses and connects directly with the spinal system of the sinus. As shown in the previous figure, it also provides the principal drainage of the cranial venous sinuses by emptying into the maxillary vein. The components of the dorsal system of sinuses. The dorsal system of sinuses comprises three principal sinuses. The dorsal sagittal sinus lies in the falx cerebri and is the principal site of arachnoid villi. It empties at the junction of the midline of the left and right transverse sinuses. The straight sinus drains the great cerebral vein into the dorsal sagittal sinus. It lies in the caudal part of the ventral edge of the falx cerebri. The transverse sinus is considered to be paired. The left and right sinuses are joined on the dorsum in line where they receive dorsal sagittal sinus. They continued laterally the connecting tissue. The ventral system contains three main sinuses. The cavernous sinus is strictly a pair of sinuses, but the pair cross-connected and can therefore be regarded as a medium sinus. Dorsal and ventral petrosal sinuses. The dorsopetral sinus drains the ventral region of the brain. The ventral petrosal sinus is buried in the phone and simply joins the cavernous sinus to the connecting sinus. Drainage of the cranial sinuses into the systemic circulation. The cranial system of venous sinuses drains mainly the other connecting sinus into the maxillary vein. Strictly, it is the transverse sinus that drains into the maxillary vein via the temporal sinus. The emissary veins drain the cranial system into the occipital vein and the internal jugular veins. The veins of the brain. The veins of the brain are unusual in having no valves. Also, they have no smooth muscle in their walls and are therefore very thin wall. and that's the most seas between the veins of the brain occur relatively commonly as compared with the arteries, particularly on the surface of the brain. Unlike the arteries, there are also some anastomosis within the substance of the brain. Since the veins have no valves, there is a possibility that alternative channels may develop if a vein is obstructed. The veins of the more caudal part of the brainstem correspond essentially to the arteries. The cerebrobates, however, are not satellites of the arteries. These veins are arranged into groups, superficial and deep. The superficial cerebral veins drain bidorsal cerebral veins into the dorsal system of cranial venous sinuses and biventral cerebral veins into the ventral system via the dorsal petrosal sinus as shown in the next figure. The deep cerebral veins drain the deep dorsal parts of the cerebral hemispheres. These veins empty into the great cerebral vein and hence into the straight sinus. Clinical significance of venous drainage of the neural axis: spread of infection in the head. Infected material which enters the veins of the face, nasal cavity, orbit, and upper teeth can drain directly into the ventral system of cranial sinuses. Bacterial attack on the endothelium of the sinuses may cause thrombus, and this can lead to sudden obstruction of venous drainage in the sinus system with serious if not fatal results. Paradoxical embolism. The continuity of the valveless sinus system throughout the whole length of the neural axis enables bacteria or tumor cells to travel from the thorax or abdomen to the head or from the head to the thorax and abdomen. For instance, abdominal straining can produce a pressure gradient within the sinus system capable of driving blood from an abdominal organ such as a kidney into the spinal sinuses. Such a flow would initially have to take place against the valves which guard the intervertebral veins connecting the venous sinuses with the caudal vena cava. However, during straining the pressure rises in these paintings and distance them, and this is evidently sufficient to render their valves incompetent. Abdominal blood can then enter the longitudinal spinal sinuses and travel to the head or drain into the vertebral vein in the neck or to the azygos vein in the thorax. In this way, microorganisms from an infected kidney can reach the head, neck, or thorax. These routes produce unexpected move action or tumor cells, a phenomenon known as paradoxical embolism. Venous obstruction. Probably because of the frequent occurrence of venous anastomosis even below the surface of the brain, lesions resulting from venous occlusion appear to be much less important clinically than those arising from arterial obstruction. Nevertheless, from general principles, it can be predicted that a sudden blockage of a substantial vena may cause capillary hemorrhage in the area which it drains. Extensive occlusion by thrombosis of a considerable length of such a vein will certainly cause severe damage to the brain. The actual recognition of lesions resulting from intracranial venous obstruction has been slow in both man and mammals. Observations have been made, but it is known that man's surgical interference with the great cerebral vein can lead to coma and muscular rigidity. It has been suggested that lesions of the veins, if they were but looked for, might turn out to be the basis of certain cerebrovascular accidents in man. There is some evidence that some of the slight but distinct neurological deficits which are found in young children to hypoxic lesions mainly in the cerebral cortex, which have arisen basically from thrombosis of the dural sinuses and the Reaper paints. Such thrombosis appear to arise from prenatal circulatory failure, which is commonly due to placental fix, especially premature detachment. There's also evidence that tearing of the venous sinuses at the junction on the fox cerebri and membranous tentorium cerebelli due to excessive distortion of the head during delivery is a common injury in infants, which can sometimes lead to fatal subdural hemorrhage into the posterior fossa. Angiography for diagnosis. The pattern of the venous sinuses and veins inside the cranial cavity is clinically important, since cerebral angiography is used to diagnose disorders of the central nervous system. Most of the intracranial sinuses and veins of the brain which have been mentioned above are visible on venograms.